Good morning to all. My topic is Golgi bodies, so we can discuss about it. Here we can see the location of Golgi body in an animal cell. It is also present in plant cell and in all cells except in prokaryotes and mature mammalian RBCs. Here, Golgi bodies is the aggregation of membrane-bound spaces. Aggregation means combination. So, Golgi bodies are the combinations of membrane-bound spaces. They occur in mainly three types, that is lamellae, vesicles and vacuoles. We will discuss about it in more detail in the morphology. And then Golgi body's main function is cellular secretion, and so it is also it's more abundant in secretory cell. It is concerned with concentration, chemical modification and packaging of synthetic products as secretory granules or droplets. Multiple sets of Golgi bodies are described as dictyosomes. Golgi bodies was first observed by Camillo Golgi. Morphology. Morphology, as told earlier, it is mainly of three kinds, that is lamellae, vesicle and vacuole. This lamellae, it is the core unit and it is the flattened sacs. The internal lumen of lamellae is called cisternae. They are the core unit as told, and they are arranged in parallel stalks or piles. Now the next one is vacuoles. They are large and scattered membrane-bound sacs and they appear closer or near to lamellae. Then we can see the third category, that is vesicle. They are not small, they are just small membrane-bound spaces and they appear mostly in groups. Next is lamellar membrane. Lamellar membrane is oriented, it has forming phase as well as maturing phase. So consider this as a lamellar membrane. So this phase will be the forming phase, that is the convex side will be the forming phase, and this side, that is the concave side, will be the maturing phase. And two phases have their own function. Forming phase receives vesicles from endoplasmic reticulum and they are oriented towards rough endoplasmic reticulum or nuclear envelope. And in maturing phase, they give out large number of secretory vesicles. We can see here they are giving out secretory vesicles and they are oriented towards the cell surface. Next, lamellar stalk. There are three types, that is cis sacs, trans sacs and medial sacs. Cis sacs, that is found in the outer phase, trans inner phase, medial in the middle phase. So these three sacs have their own functions. We can look into that. Cis sacs, they help in the phosphorylation of mannose molecules of glycoproteins. Trans sacs, they help in the addition of galactose and sialic acid to form glycoproteins. Medial sacs, that is found in the middle portion, they are helping in the removal of mannose molecules and addition of acetyl glycosaminoglycans to glycoproteins. Next, we are going to discuss about vesicles. They are of two types, that is smooth surfaced and rough surfaced. Smooth surfaced vesicles are the most commonly found, rough surfaced are less common. These rough surfaced vesicles are coated with granules or bristles and they appear more or less rough type, so they are called rough surfaced. Next is the chemical composition. Golgi bodies are chemically unstable. That is, their chemical makeup can change from time to time. In general, when we are saying, we can say that their membranes are of lipoprotein composition, laminar structure and fluid mosaic organization, and they are very rich in variety of enzymes. Two enzymes that is found in high concentration include thiamine pyrophosphatases and glycosyl transferases. Next, we are going to discuss about the biogenesis of Golgi bodies. As we know, Golgi bodies, they are constantly formed, changed, broken down and reformed. And there are three different views about the biogenesis or formation of Golgi bodies. One is of that de novo origin, then they are formed from pre-existing membranes and then from smooth endoplasmic reticulum. In this diagram, it is a model of formation of Golgi apparatus from endoplasmic reticulum. So each step, when we observe, we can clearly see how endoplasmic reticulum give rise to a Golgi body. So from A to C, it depicts the formation. We can see from rough endoplasmic reticulum, layered cisternae is formed. Then in the diagram E, we can see that secretory. Here we can see secretory vesicles are formed. Then here division of cisternae occurs, and at last Golgi bodies are formed. And last, we are discussing about the functions of Golgi bodies. As we all know, as told earlier, the main function of Golgi bodies is of cellular secretion, and that is why they are mostly found in secretory cells. So they act as chemical factories or secretory centers of the cells. Now we can discuss about their functions. They serve as an intracellular compartment for the chemical modification and transformation of substances synthesized elsewhere, and helps in the differentiation and rapid turnover of cellular membranes. Then synthesis of mucopolysaccharides, glycoproteins and glycolipids by glycosylation. Then cellular secretion of export proteins, intracellular enzymes. Then formation of many substances. Golgi bodies help in formation of many substances like zymogen, melanin granules, microbodies, primary lysosomes, acrosome that is found in the developing animal sperms, then formation of cell plate, then formation, packaging and transport of cell wall polysaccharides, removal and recycling of excess membrane. So like as in a factory, many functions are done by this Golgi bodies, and they are an important cell organelle. Thank you.